Welcome, ladies and gentlemen, to this presentation from Lifebit. My name is Torben, Vice President here at Lifebit, and together with my colleague Filippo, one of our population genomics scientists, we'll be taking you to the United Kingdom's central COVID research response and the critical role that Genomics England's groundbreaking new research environment plays in it. It truly marks a turning point in life sciences, especially with focus on genomics. What is possible today will change the world of life sciences forever. With the advent of cheaper sequencing technology, the dropping price rendering genomics all but a commodity, we have seen an incredible blossoming of genomic research around the world. Lifebit alone are tracking more than 90 population genomics projects around the world with just under 6 million participants, amounting to more than 700 million gigabytes worth of data. And this is truly driving the genomic transformation of life sciences as a whole. Many of these projects are focusing on rare diseases that affect hundreds of millions of people worldwide, or the main killers like cancers. And they do that with incredible efficacy. Already every second drug in the US is approved based on genomic research. And we have been seeing a speeding up of diagnosis time between 3 and 4 times. And now it becomes a weapon in the war against this global pandemic. As a matter of fact, genomics is central to Her Majesty's government's central response to COVID-19. All of the government branches, the NHS, the main research funding bodies, and the Genomic Consortium have come together to sequence COVID data to make it available worldwide. And they work with the leading technology firms in the space, Illumina for sequencing, AWS for the cloud, and Lifebit for the research environment. In what is truly an unprecedented cohort for any one disease, the NHS and Genomic, the consortium, are collecting and sequencing the whole genomes of 20,000 patients that had to be admitted to the ICU with COVID-19. 15,000 patients who did have COVID-19 but had mild disease. And combining that data with the world famous 100,000 cohort of Genomics England of whole genomes. And this data is then safeguarded in a groundbreaking new research environment. In a partnership between Genomics England and Lifebit, this platform makes the data available to global top-tier pharma companies and leading research organizations around the world with one common goal: to understand the underlying genetic variances in the response of patients to COVID, to understand how the virus works, to understand new targets that can then in turn help the development of vaccines and treatments to rid the world of this virus. And it brings us to this question in a technical challenge session. What are the challenges that these wonderful biobanks are facing, and how does it affect researchers? This is Fort Knox. And just like Fort Knox is safeguarding the US gold reserve and probably one of the most secure places on earth, the same has to happen for this valuable patient data. And Genomics England has been pioneering that and is pretty much the Fort Knox of genomics. And it absolutely makes the security of patient and participant data non-negotiable and its #1 principle. These requirements oftentimes hold back biobanks, though. It often means that data is siloed and difficult to access. It means that data can't be combined with other critical sources of data. And researchers more often than not see themselves restricted in which tools they can bring to this problem. And finally, in an environment like this, collaboration between researchers and organizations is all but impossible. And this has been hindering the research progress of these wonderful projects. So the solution to that, of course, we're at a AWS conference, is a cloud native one. But it is a cloud native one in conjunction with Lifebit's unique federated technology, where a research environment sits on top of Genomics England's own AWS cloud, keeping the data within Genomics England's secure environment, and the platform is merely communicating with it in a federated manner. Through this, what we call a reading library, as opposed to a lending library, data never leaves the secure environment, yet gives access to research organizations, pharma companies around the world to freely analyze this data and get these critical findings. In addition, federated technology allows the augmentation of Genomics England data by the incorporation of other data cohorts, of other biobanks, or private initiatives. And a federated cloud solution just really enables research. Other than ever before, we're now moving computation and analysis to the data and not the other way around. And this allows an analysis across disparate data cohorts, combining these different data cohorts without moving them into permanent storage. It allows for any tool to be brought in, as we'll show you later. And as a cloud solution, of course, it is the ultimate enabler of seamless collaboration. And Lifebit has been pioneering cloud-native solutions ever since its start. Our co-founders are the co-creators of workflow language Nextflow that is fantastic for scaling in the cloud. And with the funding of some of Europe's largest investors, we have been bringing our federated technology to ambitious research projects like the Personal Genome Project to do exactly this kind of public data and analysis access, as well as big pharma organizations like Janssen Pharmaceutical that do industry scale multi-omic analysis to the system, or where we're supporting Boehringer Ingelheim and GSK in fast-tracking vaccine development. As a federated technology, GEL CloudOS thus is rooted in Genomics England's very own AWS cloud, using its own EC2 and using its own S3 buckets where this most valuable data sits. And then federated on top of this sits CloudOS as the unified digital research environment. And it is built to leverage the near infinite resources of AWS and be fit to handle the nature and the volume of the data that we are facing here. The platform can thus analyze more than 10 million individuals with all of their 3 billion genotypes across millions of annotations and tens of thousands of clinical and phenotypic variables. And it gives researchers all the tools they need to analyze this data. From a powerful data browser on top of that, they can query that data in seconds, the ability to bring any analysis pipeline, interactive tools like Jupyter notebooks, and visualization interfaces. All that is needed that researchers can take this valuable data and make the most of it for the benefit of patients. And central to it all, we've solved the Fort Knox problem. Data remains in Genomics England environment. It never leaves it, and it is 100% secure. And that is the solution for every biobank around the world, but as well as every private organization. And now I would like to take you through the user journey of Adam. He is our model team leader scientist at a top pharma company, and he is tasked to research COVID to find a vaccine. So he wants to create cohorts based on phenotypes, genotypes, and medical records across all of these samples. He wants to be able to combine that data with his private pharma data and use his very own tools. And of course, he's doing that while collaborating with other researchers and keeps the cost in check. So today, in this demo, we'll take you through 5 steps. 1, how Adam and his collaborators can work together in a workspace, how they can bring their very own data, how they can bring their very own tools. And then Filippo will take you through how they can search and browse all of these cohorts to then kick off analyses and visualize the results. So this is Adam's home view of the web-based CloudOS, accessible by any browser, of course. And here Adam is currently in his own workspace. He can easily see which other workspaces he's part of and switch around these workspaces in just a click. In the same way, it is incredibly easy for him to create a new workspace where this is necessary. In this case, Adam is fine with his own COVID workspace, and he just wants to invite one of his collaborators, Peter, into the workspace. Because Adam is an admin, he can just navigate to the team dashboard and simply invite Peter by putting in his email address. Here already, Adam can decide, is Peter just a simple researcher, a member, or should he also be an admin, and set these rights here. He can further assign a cost limit to Peter, and this cost limit, and you'll see this throughout the platform, can be in 3 levels. It can be a soft limit. When it is being breached by Peter, both him and Adam receive email notifications, or strict limit, which means that Peter won't be able to do any further analysis after the current analysis is breaching his limit. And and the hardest is the hard limit, where Peter's current analysis would even be terminated to enforce his budget. And in addition to this, Adam can also set individual and granular permissions for the different buckets that Peter has access to or doesn't have access to. Well, he is satisfied with these settings and goes and invites Peter, who now gets an email notification and gets added to this workspace. Now, working together with Peter in this workspace, they of course want to collaborate on data. And that works through data sets in CloudOS. Here, Adam and Peter create a new data set, and these can be sold from various sources. It can be, this data can come from Genomics England data, of course, but it also can come from external data sources. For external data sources, Adam has 2 different options. He can import, whether it's straight up import. And in this case, we're uploading a small data set of 5 individuals with a COVID status that's been provided by Public Health England. And Filippo will later use this file to show you how users can easily incorporate it into the data browser in just a minute. And the second alternative is our unique federated technology. And here it is not about importing, it's about linking. So our federated technology, CloudOS, already curates a number of public data sets like Genome in a Bottle, 1,000 Genomes, or the Personal Genome Project. In addition to that, Adam can simply connect his own private S3 bucket. And that means he can now bring his private pharma data or any other public data source into the platform without actually moving the data into physical storage within Genomics England. And that truly enables federated analysis across these disparate cohorts. Now with the data imported, Adam and Peter, of course, want to work on tools. And they can do this either through the tools that the platform is already curating, which you have in front of you. These are a large list of curated open source best practices, like GATK, for example, which have all been verified and tested by Lifebit. In addition, he, of course, has his private workspace tools. Here, him and Peter can work together on tools, but also bring in new tools as they see fit. And they can do this in 2 different ways. They can either import workflows like an Nextflow straight from GitHub, or virtually any containerized application by importing it from Docker Hub. In this case, Adam wants to import an HLA typing pipeline. So that's actually a pipeline that is used for variant calling for highly polymorphic regions. And all he does, he navigates to GitHub, and in GitHub, he copy pastes the URL, or he copies it, and then he pastes it into this dialog. He gives it a name. He has a choice of making it available to other people in the community or doesn't. And then with 2 simple clicks, the pipeline gets imported into his private repository. And as you can see here, it is now available for him and Peter to use. And I would like to hand over to Filippo, who is one of our population genomics scientists and bioinformaticians, to take you through how Adam can easily use the Cohort browser to browse all of this large-scale data and then kick off both pipeline as well as interactive analysis and visualize the results.
Thanks, Torben. I will now show you and give you an overview of the Cohort browser in CloudOS and how researchers can perform all the steps of any genomic and multiomic analysis from a single platform. I will show you how researchers can visualize genomic, clinical, and phenotypic data to identify patients of interest, how they can run both batch and interactive analysis, and finally, how they can visualize and interpret the results. First, I will show you how Adam, our user persona, can easily identify patients and individuals of interest. Let's imagine that Adam, our researcher at a large pharma, has identified a group of patients at high risk of COVID infection. He now wants to replicate the same cohort with the Genomics England data and finally run federated and joint analysis in the cloud. Adam can do the first step of identifying the patients from the Cohort browser. From here, Adam can either load a previously saved cohort or simply create a new cohort by providing a name and a description. The platform has already ingested information for more than 100,000 participants with tens of thousands of phenotypic and clinical data and billions of genomic markers. For data security reason, of course, we cannot show the real data, so instead we will show some synthetic data. So bear in mind that if the distribution will look funny. Once entering the Cohort browser, Adam can see three sections. The phenotypic section, from which Adam can visualize and filter for the phenotypes and clinical data of interest. The sample section, which contains all the individuals he has identified and all their clinical information. And finally, the genotypic section, which includes all of the markers, all the 3 billion markers ingested in the platform. Adam can easily create his cohort by starting to add filters to the main dashboard. Adam can either browse for particular filters or select ones, such as the ICD-10 or the International Code of Disease. Once selecting a phenotype, Adam can see all the metadata, such as a description of the phenotype, external resources, or multiple time points if present. Moreover, Adam can also view the preview of the data. All this information is essential for Adam to understand on how to work with the data. Now, Adam wants to replicate the same cohort he has in his large pharmaceutical company, and he wants to identify patients with a coronavirus infection born before 1960 and who have been diagnosed with asthma by their doctors. To do this, Adam can simply add two more phenotypes to the main dashboard. First, year of birth, and the second phenotype, the doctor diagnosis of asthma. Now that all the information is in the main dashboard, Adam can simply select the criteria of interest, such as coronavirus infection. In this case, you can select the particular disease, and select patients with a diagnosis of asthma and born before 1960. Now you can simply apply the filter. In a matter of seconds, the platform has queried more than 100,000 participants and has returned the results. The speed of the platform and the scalability, thanks to the cloud, is essential for Adam, as it allows him to drastically reduce the time to discover process, which translates in faster research. Of course, Adam wants to incorporate more data in this cohort, and he can simply do it by providing two files. The first file contains the information and the clinical data for each patient, whereas the second file contains all the metadata, such as description and external resources. So Adam can simply load the COVID data that Torben has uploaded before. And again, in few seconds, the data will be ingested. Now that Adam is happy with this cohort, he can simply save it. All the cohorts and all the data are available to all of Adam's collaborators, effectively meaning a real-time collaboration. Now, while Adam was working with this data in the large pharmaceutical company, he has identified some pathogenic markers on chromosome 22, which are important for COVID infection. He now wants to find the same markers and filter his individuals for this genomic information. Adam can simply go to the genotypic filtering section, select the criteria of interest, such as chromosome 22 and pathogenic variants. He can filter the table, select the marker of interest, and apply it to the cohort. Again, in a few seconds, the platform will have queried and filtered for this information. As a researcher myself, I know that the importance of understanding the biological and clinical implication of any genomic information. This is why, from this single platform, we've annotated all the thousands and billions of variants against the major biologically and clinically relevant databases. This drastically enriches Adam's experience, as you can gather all the genomic information in a single platform. This is especially important for pharmaceutical company, as researcher can instantaneously understand the biological and clinical implication of their findings, which will guide drug and vaccine development. Thanks to our cloud-native approach, we are now scaling and adding more annotations to cover all of the different researcher use cases. Once Adam is happy with this new filtering, he can save again his cohort. Again, the cohort will be available to all of Adam's collaborators. Directly from within the Jupyter, from within the Cohort browser, Adam can launch interactive and batch analysis. For interactive analysis, Adam will be redirected to a Jupyter session. From here, Adam can choose the best instance for his analysis. Adam has access to the entire AWS computer range, and he can filter instances by price, CPUs, or memory. Adam can choose between general purpose instances or more specialized ones such as GPU and FPGAs. When Adam needs clusters, for example for Spark, Adam can filter for instances with EMR bolt-ons. Also, when required by his analysis, Adam can freely assign elastic block storage to his instance. More importantly, Adam can switch between instances in his Jupyter session, meaning that he can accommodate his analysis and his workload very efficiently. Once Adam is happy with all his parameters, he can simply start a new Jupyter session. Once entering in a Jupyter session, Adam can launch notebooks in the most commonly used languages, such as Python, Julia, and R. Most importantly, and thanks to the cloud, all the Jupyter sessions, together with all the data associated with them and all the codes, are shared with all of Adam's colleagues. Again, meaning instant collaboration and real-time collaboration. From within a Jupyter session, Adam can also easily add more data or notebooks. In this case, I will simply load the notebook that Adam has used for his own COVID research. Adam can also launch batch analysis in 3 simple steps. First, you can select a pipeline of interest, such as a GWAS. As a second step, Adam provides all the data and parameters. For the sake of the demonstration, I will simply use the example data. CloudOS supports full versioning, meaning that Adam can choose which version of the tool he wants to use for his analysis. Once Adam is happy, he can simply go on next and on the third and final step. From here, Adam can choose, again, the best instance for his analysis. In case of batch analysis, Adam can also access the entire AWS EC2 range of spot instances. And again, you can add EBS storage as required. Adam can also add the cost limit and the limit type, as he can do in a Jupyter session. Finally, once Adam chooses spot instances, he can mark the job as resumable, meaning that if the instance is terminated, the analysis will restart from the latest step. Once Adam is happy with the instance and with the summary of the pipeline and the input data, he can simply run the analysis. Adam will now be redirected to the main pipeline job, from which you can see all the analysis he has run, their status, but most importantly, the runtime, the cost, and the resources being used. Once entering a pipeline analysis, Adam can see all the parameters and containers, as well as a breakdown of all the resources by sub-process. One of the most important steps of any genomic analysis is to visualize actionable data, and Adam can do so from the multi-QC. The multi-QC provides specific visualization for each kind of analysis. In the case of a GWAS, Adam will see a Manhattan, volcano, and QQ-plot, which are fully interactive and annotated. Adam can also see the top results from his analysis directly from the report tab. And again, all the variants are annotated against the most clinically relevant and biologically relevant databases. This effectively means that Adam can instantaneously understand the clinical and biological implication of his finding. It is important to stress that CloudOS fully integrates with the client AWS, being the client a large pharmaceutical company or, such as in this case, Genomics England. This means that the analysis is run in the client S3 bucket, and the data never leaves their environment. This approach is radically different from all the legacy systems in which researchers had to move data outside of their S3 bucket. CloudOS also integrates with other visualization tools, such as the IGV Genome Browser, which are widely used by the research community. As a cloud operating system, Adam can perform programmatically via API and command line interface all the different activities I've shown you, from running analysis to fetching the result and identifying individual patients of interest. To conclude, I've shown you the Cohort browser in CloudOS and how researchers can perform any end-to-end genomic and multi-omic analysis. From importing data and tools to identify the right patient for their analysis, until running any batch or interactive analysis and visualize and interpret the result. I will now pass it back to Torben, who will conclude the session.
Thank you very much, Filippo. And now to conclude, life sciences going forward will truly be different in the space of genomics. We've essentially taken Genomics England, the pioneers of population genomics, and have turned them into the world's most cutting-edge research environment. And together with this incredible COVID data that the UK is making available to the world, we can bring this data into the hands of researchers around the world for that impact that we are all praying for, that we're all needing. And it is done in the most flexible of ways. Bring your own tools, bring your own data, all of the things that bleeding edge of innovation has as a prerequisite. You cannot can research tools. There are thousands coming out every single year, and researchers need to have full access to those, as well as all of the data they can get. So what we've in essence done, we've taken the Fort Knox of genomics, that is Genomics England, and turned it into the International Space Station. Security for data, but now high tech for researchers available at their fingertips. And the federated technology of Lifebit that is making all of this possible is also paving the way into a world where data doesn't have to exist by itself, a world where data doesn't have to move, but we're able to run analysis across different data sets, different biobanks, private data, to achieve the most out of this data. Because together, data is worth more than the sum of its individual parts. And this platform is now available to every AWS customer in their very own environment, to every biobank around the world, public or private, to every research institution, and to every pharma and biotech organization, to have the same scope and the same cutting edge of research done in their own environment, on their own data. And with that, I'd like to thank you for your attention.